Okay. So today we're going to continue our discussion of arterioles. So when we left off, we had been talking about local regulation of arterioles, particularly thinking about the relationship between flow, pressure, and resistance, and how we see changes in the state of the smooth muscle, constriction and dilation, that adjust the amount of flow to different organs, different capillary beds. We had started to think about how this can be flipped around a little bit to think about relationships to blood pressure or our mean arterial pressure. So when we left off, I think we were somewhere around this slide, beginning this idea of extrinsic control. So here's our reminder that when we're thinking about blood flow, we're thinking about a relationship where a difference in pressure or perfusion pressure is necessary to power flow moving to an organ or to the organ system. When we have resistance, often due to small radii of vessels, that resistance resists the flow, can drop flow down a little bit. We know we have physiological variables that we can sub in for all of those factors now. So when we're thinking about flow to our systemic circuit, we're really thinking about the cardiac output. When we're thinking about a difference in pressure, there's very low pressure in the veins coming back to the heart. So we can mostly ignore them and just put in our mean arterial pressure as our big power for movement of blood through the system, divided by our total peripheral resistance. So resistance anywhere and everywhere in all the little blood vessels throughout your body, giving us that cardiac output. And when we rearrange that equation, so multiply the cardiac output by TPR, that gives us the relationship between blood pressure. So we've isolated blood pressure, this mean arterial pressure. Now we know that this is related to our cardiac output and our total peripheral resistance. We can just multiply them together. So as we move forward and kind of build towards regulation of blood pressure, this will become important. So we also want to break that down a little bit further. We'll talk about this extensively when we talk about regulation of MAP. I'm just going to point out again that we know that cardiac output is our stroke volume times our heart rate. All right, so anytime we had the heart squeeze harder, we increased cardiac output. Anytime we had more heartbeats per minute, we increased cardiac output. So those can then be placed into this equation right here. So now we know that if we increase stroke volume, we would increase mean arterial pressure. If we increase heart rate, we'd increase mean arterial pressure. And if we increase TPR, our total peripheral resistance, you're going to tell me what's happening to that mean arterial pressure. I'm going to go ahead and click it through. Okay. So it should still be raising our mean arterial pressure, right? We're just multiplying across to get that mean arterial pressure in our rearranged equation. So to do that, right, we said stroke volume goes up, MAP would go up, heart rate goes up, MAP would go up. And now we also know that if TPR goes up, MAP would go up. So lots of ways we can raise mean arterial pressure. Dropping down any of those factors would have the opposite effect, dropping mean arterial pressure, dropping your blood pressure. So that's going to come into play as we think about this extrinsic control. The other thing I'm going to remind us of as we move into extrinsic control is about our autonomic nervous system. So when we think about regulating extrinsically, so with hormones, with nerves, the smooth muscle and our blood vessels, this is something you don't think about. So this is under autonomic control, so that kind of automatic function of our nervous system that does stuff without you particularly noticing unless something's going real weird. Okay, so we had our sympathetic and our parasympathetic branches, and we're going to be thinking in particular about the sympathetic half of the autonomic nervous system as we begin to think about extrinsic regulation of our arterioles. Okay. So a reminder, these were our three roots. This is a picture over all of your sympathetic nervous system. So we had a couple different ways that we can have our arrangement of preganglionic and postganglionic neurons. All right, so our 1-2 chain going through the peripheral nervous system out to our effector organs. Okay. But overall, what I want to key in on here, we know that in general, we're going to be releasing norepinephrine from our postganglionic neurons or our adrenergic neurons because they're releasing norepinephrine at the end, or epinephrine if we're going through this route where we're specifically activating that inside part of the adrenal gland, the adrenal medulla. Okay, so norepinephrine and epinephrine, also known as noradrenaline and adrenaline, which is where we get that adrenergic word from. Yeah. So we're going to be thinking about these neurotransmitters and hormones as we think about regulation of the smooth muscle around the arterioles from the kind of central command center. Okay. So when we're thinking about extrinsic control, our intrinsic control, we were thinking either about diffusion of gases locally having an effect when we thought about active and reactive hyperemia, or we were thinking about the myogenic response, where we basically have smooth muscle that, when stretched out, kind of bounces back, trying to keep flow constant. Now that we're thinking about extrinsic control, we're thinking about signals from far away that have an effect on the smooth muscle in the arterioles. So that means we're thinking about nerves, we're thinking about hormones. And particularly, we're thinking about sympathetic activity when we're thinking about the nervous side, which means that we're going to be thinking about norepinephrine, and we're going to be thinking about our hormone epinephrine. Okay. So hopefully, we remember a little bit about our different receptor types. If we don't, we're going to get a reminder. So you're going to dig out, thinking in your minds about different types of receptors we had specifically for norepinephrine in this case. You need to tell me the name of a receptor that likes norepinephrine, has an excitatory effect on effector organs, and internally in a cell would involve our signaling molecule IP3. What is the name of basically the class of receptor I'm referring to here? Okay, take another second, pick a choice, and we'll look through. And then we'll jog our memories overall on receptors. Take a look. This is actually an alpha adrenergic receptor. Okay, so good job on the adrenergic part. All right, so adrenergic referring to the adrenal glands, referring to adrenaline associated with the sympathetic nervous system and adrenal function. We had two main classes, our alpha and our beta types. So I'm going to be pulling up repeatedly this table from your book, this handy dandy kind of cheat sheet. All right. But reminder, we're trying to tell apart alpha and beta receptors, one of the big things we're looking for is the relationship to this internal transduction mechanism we can think about. I'm circling this because every single one of those beta receptors activates cyclic AMP. So if I give you a signal transduction mechanism that it does not involve activating cyclic AMP, it is not a beta receptor, not a beta adrenergic receptor. So specifically, I was describing to us this alpha-1 receptor here. Okay. Other ways we can think about this, right? If we're looking for alpha receptors, we're going to notice both of those alpha receptors prefer norepinephrine to epinephrine. And that's going to come into play here as we start to think about smooth muscle and our arterioles. And you'll notice we didn't focus too much on the effector organs when we spoke about these last time because we were focusing on the nervous system part. But we were learning the different types because now we're going to be thinking about the effects on blood vessels specifically. So you'll notice here that we have alpha-1 receptors in our vascular smooth muscle, right? So in the smooth muscle of our blood vessels. We have some alpha-2 receptors too. You can see vascular smooth muscle here as well. We can see that the other place we have blood vessels listed in particular are these beta-2 receptors are going to be in some, but not all of our blood vessels. So this is why we're jogging our memory on this, because as we start to think about how our blood vessels going to different parts of the body react to the sympathetic nervous system, to norepinephrine and epinephrine, where we see these different receptor types is going to matter for the distribution of blood flow under kind of stressful conditions. Because remember, we're thinking sympathetic nervous system, we're thinking fight or flight, we're thinking stress. Sympathetic and stress both start with that S. Okay. That's our brief reminder as we start to think about how the sympathetic nervous system is controlling the arterioles. Okay. Reminder, when we're thinking about controlling the arterioles, we're really still thinking about dilation and constriction. Okay. So what happens usually with the sympathetic control of arteriolar radius is that most of our arterioles have alpha receptors, right? So most of them have those alpha adrenergic receptors, right? That we just said prefer norepinephrine, right? So norepinephrine has a higher affinity for those alpha receptors. Alpha receptors have higher affinity for norepinephrine. That's what they prefer, okay? So anytime you have norepinephrine binding to an alpha receptor for our purposes, this causes vasoconstriction. So if we have norepinephrine binding to an alpha receptor, that alpha receptor creates constriction, vasoconstriction in the smooth muscle, right? So now we've got a smaller radius for our arteriole, which means we have higher resistance. We know that TPR is just the resistance added up throughout your whole body. So overall, the effect of the sympathetic nervous system activating alpha receptors is going to be to increase total peripheral resistance and to increase mean arterial blood pressure. So we're basically increasing blood pressure overall when we have this sympathetic nervous system active. And that's because of these alpha receptors. However, okay, so that's going to be true if you're thinking, we're going to have some imaginary cells. Okay, so random organ. This will be like a liver cell or something, right? But we're going to have to pay special attention if we're thinking about, say, a heart cell or skeletal muscle cell, right? So this will still be a cell. I'll make this a heart cell. Okay. So we have our alpha receptors kind of everywhere, right? So we saw, we had alpha receptors here in this random cell. So we can put our liver cell as having an alpha receptor. We'll still have some alpha receptors here on our heart cell. That's our stand-in for skeletal and cardiac muscle. But we also have something else. So if we're thinking about a heart muscle cell or a cardiac muscle cell, the arterioles going to those cells are going to have beta-2 adrenergic receptors on them, lots of them, right? So we'll still have a little bit of an alpha receptor, but we're going to have lots of beta-2 all over the place. Okay. So this means that if we're thinking about the blood flow in the arterioles going to skeletal muscle, so our named conscious control muscles, or our heart, to the muscle itself that's beating, we're going to have a different reaction to activation of the sympathetic nervous system in those organ systems specifically. So your heart and your skeletal muscle react differently when the sympathetic nervous system kicks in than your liver does, than your GI tract does, than random other types of tissue do. Okay. So this is particularly going to be relying on these differences in the affinities of these different receptors for different hormones and neurotransmitters. Okay. So we said those alpha receptors prefer norepinephrine. And what we'll notice about these beta-2 receptors is that they highly prefer epinephrine. Okay. So an alpha receptor, basically, whenever we have something bind to an alpha receptor, the effect is going to be vasoconstriction. Right? So we just spoke on the previous slide that when we have norepinephrine bind to those alpha receptors, as they prefer, we constrict the blood vessels down. You'll notice I'm going to jump down a bullet point here. But when we think about alpha receptors and epinephrine, right? Epinephrine is less preferred by those alpha receptors, but they will still bind to it if there's a lot of it, right? So if we have epinephrine binding to an alpha receptor, it's still going to be dependent on the receptor type, basically, the reaction. So that's still going to be vasoconstriction at this alpha receptor. Sorry, my scribble's going back and forth here. But if we're triggering those beta-2 receptors, really the way we'll trigger a beta-2 receptor is with epinephrine. And if we trigger a beta-2 receptor, the effect is going to be vasodilation. Okay. So what happens here, basically, is that the epinephrine, when it kicks in, when we see epinephrine in our system and comes and touches those beta-2 receptors in the heart and the skeletal muscle, that's going to cause dilation. We're going to have extra blood flow, essentially, to the heart, to the skeletal muscle. And then our alpha receptors are going to kick in at some point and start to vasoconstrict, creating an increase in blood pressure and kind of maintaining our blood pressure. So we're going to have kind of like a 1-2 punch going on as we activate the sympathetic nervous system. Cool. So the effects of epinephrine are going to be concentration dependent. Now, when we say concentration dependent, how much epinephrine you have in your system is essentially going to tell you which receptors are able to soak up that epinephrine. So I'm not saying that epinephrine itself is different, right? The epinephrine is just the epinephrine. But if you only have a little bit of epinephrine, those beta-2 receptors hog it all up. So as you start producing epinephrine, those beta-2 receptors love epinephrine, right? So they're going to soak up the low concentration of epinephrine, right? At those low concentrations, the alpha receptors don't care that much about epinephrine because they'd rather have norepinephrine anyway, right? So at these low concentrations, as we start to have epinephrine in our system, what we're doing is we're binding to beta-2 receptors in the heart and skeletal muscle. We're seeing vasodilation, right? So radius of the arteriole gets bigger, resistance drops. So we essentially see more blood flow to those areas. Okay. But this does create a problem because overall, as we start to have vasodilation happening, right, we know that we also have this relationship between mean arterial pressure and our cardiac output, which I'll write as stroke volume times heart rate. So we always have all of it there. Okay. and our total peripheral resistance, right? Okay. So once we start to vasodilate, right, so expand our arterioles, right? We had epinephrine, we bound it to those beta-2 receptors. We started to have dilation, means the resistance in those blood vessel beds went down, right? Total peripheral resistance is just the total resistance, right? So we added it all up. So what we've started to do, this is a problem we're going to kind of want to fix. We've started to drop down total peripheral resistance, which means that we're starting to kind of drop our blood pressure, right? So if nothing else happened at this point, as we route more of the blood to the heart and the skeletal muscle, that would be great. We get some more flow there, but overall our blood pressure would be dropping, which is not great because you still want oxygen, glucose going to your brain, going to all the organs that need it, not just to the heart and skeletal muscle. So this is why it's super helpful that as we see increasing concentrations of epinephrine, right? Increasing concentrations of epinephrine start to be able to bind to those alpha receptors that we have everywhere else, right? So when we have more epinephrine, we can then also bind the alpha receptors, okay? And as we start to bind the alpha receptors, if you bind to an alpha receptor in smooth muscle, has the opposite effect that those beta-2 receptors did, okay? So when we have enough epinephrine, when we have more epinephrine, we start to flood our system. There's tons and tons of epinephrine. It's going to bind to some alpha receptors. That's going to cause constriction, which means that we are lowering a radius, we're lowering a radius, therefore raising resistance. So we're pulling that blood pressure back up, right? So we're pulling TPR back up with those alpha receptors. All right. Oh, I need the pen. There we go. All right. So alpha receptors binding to epinephrine pull total peripheral resistance up, which overall is going to pull our mean arterial pressure up, kind of maintaining that blood pressure and making up for the fact that that dilation to the heart and to the skeletal muscle is starting to kind of pull our blood pressure down. Yeah. Now, if we step back and kind of think about this as a whole, this kind of makes sense if you think about what you would be doing, right? So you're in a stressful situation, you're running away from something, right? So you want to be able to fuel the heart so that you can contract more, so that you can raise your stroke volume, so that you can raise your heart rate. You're going to produce lots of ATP to do those things. You need more blood flow, right? So your beta-2 receptors, as we start to kick in that epinephrine, let you have more blood flow to the muscle of your heart. Same thing for your skeletal muscle, right? As you're running away, you're going to be burning energy. You want lots of blood flow, lots of oxygen, lots of sugars running to the skeletal muscle, right? But if we are just dilating blood vessels, if we're just dilating blood vessels to the heart and to the skeletal muscle, you'd like faint, basically, right? Because your blood pressure would drop down. That would do you no good. You'd start running, then fall over. That's not helpful. Okay. So that's why it's particularly convenient and necessary that we keep our blood pressure elevated. We keep our mean arterial pressure up by constricting the blood flow going to all the other parts of your body. So you're going to constrict blood going anywhere that's not the heart, the skeletal muscle, or the brain. We don't constrict blood vessels going to the brain. So you're not going to have alpha receptors in the brain, but everywhere else you kind of got some. Okay. Questions about this process? I think it is most helpful to think about it as each receptor type having an effect on smooth muscle, and then secondly, to think about what triggers that receptor. But I recommend thinking it through and trying it in a couple of different ways as you're practicing. I'll ask us some review questions on this next week because hopefully you've all read ahead and so you have no questions because this is all very familiar and you're a little bored. But I think the first time you see this, it can kind of be a mess of letters and numbers. So we'll do some more practicing. So other things are happening as you are exercising as well. So we're thinking about the relationship between these receptors, the hormones and neurotransmitters as our sympathetic nervous system is activating. It is also true that our cardiac output would be going up, right? So we know that the autonomic nervous system isn't just regulating blood vessels, it's also physically regulating the heart. We know that both parasympathetics and sympathetics affect the heart rhythm, right? So sympathetics make your heart beat faster. That's what's relevant here. Parasympathetics will kind of slow you down. We know that the sympathetic nervous system also contributes to the innervation of the ventricles. So we also know that when the sympathetic nervous system is active, stroke volume goes up because you squeeze harder, basically. Okay. So we do have kind of all our variables changing a little bit as we are exercising. Okay. So that's also true. Now we're tacking on that we know that the receptor types present in the heart and the skeletal muscle are making sure that as we are increasing how hard the heart is contracting and how fast it's contracting, we're getting sufficient blood flow to the muscle itself to be able to have enough energy to actually do that. And in order to keep our blood pressure from dropping, we're kind of squeezing down, constricting blood vessels elsewhere in the body, and particularly in places like the GI tract and the kidneys. We're going to be decreasing blood flow to other organs so that that blood can go to the heart and the skeletal muscle, and also so that your blood pressure doesn't drop and you don't faint. Okay. So we're disproportionately diverting the flow to our muscles. So that extra cardiac output, pretty much all of it, almost all of it, is going to the muscles, and we're contracting down everywhere else in the body. So we saw a figure earlier on Wednesday, I guess was the last time I saw you, of what that distribution looks like of blood flow when you're at rest versus when you're exercising. So if you're curious on the exact numbers, you can take a look back at that. That's the big one that we want to understand for extrinsic control of arterial radius, and therefore partially regulation of our mean arterial pressure. Right? We want to think about sympathetics, the fact that they can trigger both alpha and beta receptors. When they trigger alpha receptors, we're constricting. When we trigger those beta 2 receptors, we're dilating. Overall, because this is concentration dependent, overall, we manage to keep our blood pressure up, okay, with this kind of combined control. But we do have other hormones involved. Okay. So we already kind of mentioned the epinephrine because that goes along with our sympathetic nervous system because of that relationship to the adrenal gland. Okay. So when we see that epinephrine, most places in the body, most arterioles out in the body, that's going to, if it's high enough concentration, bind to an alpha receptor and constrict. We know that if it's specifically contacting arterioles going to heart muscle or skeletal muscle, it would hit a beta 2 receptor and dilate blood flow there. Other hormones that we will talk about in upcoming units can also play a role in control of the smooth muscle surrounding arterioles. So we'll talk about vasopressin or ADH a fair bit when we talk about the kidneys themselves and filtering blood. Okay. So that's a hormone that we mentioned once briefly before when we were talking about the pituitary. Right? We had about a gazillion hormones coming from the anterior pituitary. ADH was one of 2 hormones we had secreted by the posterior pituitary. Its job is going to have to do with regulating like blood volume and therefore also kind of sideways regulating blood pressure. So when we see that hormone kicking in our system, overall, this is going to be a way that we keep blood pressure up. So we're going to see vasoconstriction happening at the same time as we have the body pulling extra water into your blood vessels, pulling blood pressure up. Okay. Another hormone we'll see very briefly in the future is something called angiotensin 2. It basically works the same way. So fortunately, both of our hormones that we're thinking about here, the vasopressin and the angiotensin 2, we're going to be thinking as increasing vasoconstriction, therefore increasing total peripheral resistance, ultimately kind of increasing your blood pressure. Okay. So we're now going to move one smaller when we're thinking about blood vessels. Okay. So before we link everything together into our full discussion of regulation of blood pressure, we're going to be thinking about all of our blood vessels, right? So we had the heart, where we started. We had our aorta and our arteries coming out of it. We've talked about those arteries. Big thing about the arteries was they were a pressure reservoir. They had that low compliance, right? So we had arteries. Now we've talked about arterioles, right? So our arterioles are a little smaller, right? Their big thing is resistance and the ability to vary that resistance. Right, so we're thinking about pressure with arteries. We're thinking a lot about TPR and resistance as we think about arterioles. Now moving forward, we're going to be thinking about our little capillaries, our capillary beds. Okay. And the big thing that we're going to be thinking as we think about capillaries is exchange. We're going to be thinking about exchange of number of things get exchanged across the capillaries, but our big thing that we're going to think about for this unit is fluid moving across capillary walls. And then we'll return through the venules and veins later on. Okay. So we're going to start by thinking about what a capillary looks like. So we're going to learn a little bit about what the walls of that capillary look like, how they're arranged, and we're going to think about how we get blood flow going through the capillaries, leading up to our big topic, movement of material across capillary walls and something called net filtration pressure. So if you haven't looked through that yet, please do so this weekend. So NFP is going to be a big topic that we'll be focusing on. Maybe we'll get to a little bit of it today, but it'll be a focus early next week for sure. Okay, cool. So our big overarching question for our capillaries is we're trying to understand movement across those capillary walls. And we're thinking about diffusion. We're thinking about what the drivers of diffusion are and why things are able to move. So capillaries are specialized for exchange between blood and tissue, which is why they're so small, right? So they're 5 to 10 millimeters in diameter. They they can get quite small. They can get very tiny. And we're going to see that their walls in particular are very thin. So we only really have one cell layer if we're thinking about the wall of a capillary, which is a really small barrier to diffusion, right? So diffusion, reminder, that's a form of passive transport, where we're just moving down a gradient if we're thinking about something that's nonpolar, if we're not thinking about an ion, or with our electrochemical driving force if we were thinking about ions, okay? But we still have to move through physical space, right? So we want a really small physical space to have to move across. So it's really handy that we only have to move across one cell layer for capillaries, okay? Now, capillaries themselves individually are really small, but there are tons of them. So if you lined them all up and took a total cross section through all your capillaries, this is actually a really big area. So your blood is hanging out in capillaries for kind of a long time. We kind of have slow blood flow through these capillaries because we have so many routes for the blood, right? We've split so many times, right? We've divided everything up so much that we can have relatively slow movement through any particular individual capillary, which means we have enough time for stuff to cross that wall, for stuff to go back and forth, for exchange to happen. You have about 10 to 40 billion of them in your body. Their total surface area would be like 600 meters square. Almost all your cells are within a millimeter of a capillary. They're about a millimeter long end to end. Right? This is handy. You want your cells close to a capillary because your capillary is bringing oxygen, it's bringing glucose, it's taking away carbon dioxide. So this is handy. Okay? And when we look at the lining of a capillary, so we're going to have just one cell layer really forming the wall of a capillary. We're going to have some pores, some gaps in that wall even. So our endothelial cells, endothelial is the inner lining of our vessel. We've got some pores and the plasma part of your blood, right? So not the cells part, but all that extra kind of fluid in it, right? Our plasma was one of our types of extracellular fluid. That plasma can pretty freely move through the pores, although any larger proteins, any larger components in the blood are not going to be able to move across that wall. Okay. When I say a capillary bed, I've said this a couple of times, probably without defining it. What I'm talking about is just a network of capillaries. They get big. There are lots of them. They're all really small. So usually when we're thinking about capillaries, we're thinking about a collection of them. We're thinking about a capillary bed in a specific location, in a specific organ, rather than focusing on one tiny individual one at once. Okay. So here we see the relationship between the total area that the blood vessels take up and how fast blood flows through those blood vessels. So you can see here, we didn't have all that many aortas, right? We just have the one aorta, though we name different parts of it in different parts of the body. We have a couple more named arteries. We have a bit more branching, but relatively few in number. We know that in those arteries, blood is going fast, blood is going hard. We've got lots of pressure, right? Okay. As we start to split further down, right? We're in our microcirculation, we're in our arterioles. We have more branching, right? So the area is going up and the speed at which our blood is going through starts dropping down. Once we're in our capillaries where we have tons and tons of area, that's going to be the slowest that the blood is flowing as it goes into all those tiny little side routes. Okay. And we're going to pick up as we start to recombine our little tributaries into venules and then into veins. So if we're thinking about a capillary, we'll have a couple different types. Okay. So our continuous capillaries are the most common. So the continuous capillaries are usually what we're talking about. We have our endothelial lining, and we have relatively small gaps between the cells. So I'm drawing endothelial cells kind of poorly, right? We're going to have small gaps for things to pass through, not that much space. So we'll have some small water-soluble molecules able to move through gaps sometimes, but definitely not big proteins. If we have a fenestrated capillary, a fenestra is a window, we're going to be thinking about huge gaps between those endothelial cells. So a fenestrated capillary is going to have even more exchange happening across it. Okay. So if you had a fenestrated capillary, you could have some proteins, potentially some blood cells moving into the blood in those areas where you have a fenestrated capillary, which is good. We have to make blood cells somewhere. We have to get them into the bloodstream somehow. So fenestrated capillaries have their uses. Usually we'll be thinking about continuous capillaries. There are some other types of capillaries, discontinuous capillaries and sinusoids. We won't really be thinking about those. Okay. So capillaries, like the arterioles, are part of the microcirculation. So you're not going to see them physically in the lab, although I was just staring at my face in the mirror. You might occasionally see a capillary if you have like ones that burst on your skin. So if you see little red lines across your skin, that might be a burst capillary. But if they're normal, you're not going to see those capillaries. Okay? You're also not going to see your arterioles, your meta-arterioles, or your precapillary sphincters. Okay, so when we're thinking about the circulation, thinking about the arterioles going to the capillary beds. But we're also going to want to be able to kind of control flow through a capillary bed and through specific parts of capillary beds, which is where these meta-arterioles and precapillary sphincters are going to come into play. Okay, so here's what we're looking at. So here we have our blood flow coming from the heart, and this is actually starting at the level of an arteriole. So this big one over here is the arteriole passing by our capillary bed. So this whole network coming in the middle is what I'd usually be referring to when I'm referring to a capillary bed. On the other side, after blood travels through the capillary bed, it's going to return to the heart, first through our venule, and then ultimately into a vein, ultimately into the vena cava, back to the heart. Okay. Now, sometimes we're going to want to route blood kind of past a capillary bed. We're going to kind of want to skip a capillary bed. Sometimes we might want to force blood to go through specific parts of a capillary bed. So one of the ways we can do that is with our meta-arterioles and our precapillary sphincters. All right, so we can regulate a little bit with the arteriole or smooth muscle, how much resistance we have in that vessel would overall bring more or less blood to the capillary bed as a whole. The meta-arteriole here, basically a bypass, like a highway bypass, through the center of this capillary bed. Okay. So a meta-arteriole, kind of a shunt, is another way we could think of it, that allows us to kind of skip going through the capillary beds if we don't want to right now. Right. So when we have our meta-arteriole, our big meta-arteriole open here in the middle, we're going to have a lot of blood, comes down the arteriole, heads into the meta-arteriole, doesn't bother going through these little local roads on the side here, goes straight to the venule, back to the veins, and mostly kind of skips over these capillary beds. However, our meta-arterioles are not always open. Okay, so the meta-arterioles do have rings of smooth muscle at strategic locations, so they can essentially contract and expand. We don't have all that many of them drawn on. I wish this one wasn't drawn exactly right here, but you can imagine little bits of smooth muscle in that meta-arteriole. Okay, so we're going to have it at strategic locations. Okay, if we contract down our meta-arteriole, right? When you contract a meta-arteriole, when you contract muscle, vasoconstriction, decreasing radius, increasing resistance, therefore kind of lowering flow. So you can think of contracting this meta-arteriole as kind of blocking passage through the meta-arteriole. Okay. So if you contract the muscle controlling that meta-arteriole, start to kind of block it off, this is going to force blood to go around it. So essentially we'd be forcing blood through into our capillaries. Now, when we relax the muscle controlling the meta-arteriole, we open it up, we dilate it. If we've got a big passage right here, right? Our meta-arteriole has a larger radius, larger diameter. It's wider than the capillaries. So all else being equal, it has lower resistance. So more blood is going to go through the meta-arteriole and kind of skip over the capillaries if it's open, if we have that muscle kind of relaxed around the meta-arteriole. We also have precapillary sphincters. So they're a different thing. So the meta-arteriole lets us bypass the capillary bed when it's open. The precapillary sphincter is kind of like these seals on the beginning of the capillaries coming off of that arteriole and meta-arteriole. So this is again a ring of smooth muscle, contracts and responds just to local factors. So things like that oxygen and CO2 concentration we were thinking about last time. That's the kind of thing you want to be thinking about here. So not nerves, not hormones. Okay. Now, if we have this precapillary sphincter contract, right, that's going to make our entrance to the capillary smaller. So smaller radius, larger resistance, less flow. So if this one squeezes down, this one is labeled in a convenient place, drawn in a convenient place. If this starts to constrict down, right, we're going to essentially be blocking it. We're going to have less flow through that capillary. If it relaxes, right, if it relaxes, opens up, right, we'd be lowering the resistance. So now we'd have more flow through that part of the capillary bed. When we think about what is causing this contraction and relaxation, metabolites cause relaxation. So we mentioned metabolites. So we're thinking about byproducts of metabolism, like we were thinking about CO2 when we were thinking about hyperemia. There are other metabolites that are involved in these kind of regulatory processes, but they work in kind of the same way. Okay. So what we're leading up to, right? So we know that we've got lots of capillaries, lots of area. We've got slow flow through that large area. And we know we can kind of control flow through that area. But our big picture is we're going to be trying to get exchange going on across the capillary walls. Right. So we mentioned the diffusion. A type of passive transport is our most common mechanism of having stuff move into and out of our capillary. Depending on what we have moving, it's going to move a little bit differently, right? So if we have something that is lipophilic, which is a word we may or may not have seen before, right? So lipo is for lipid, philic, it's for loving. That's in Greek, like a love, right? So we love lipids. Remember that our cell membranes are primarily made of phospholipids. So stuff that likes lipids is going to be able to just directly diffuse across a membrane. Right? So we thought about things like our small nonpolar gases, oxygen and carbon dioxide when we think about that. There are things like certain hormones and stuff that might also go in that class, certain vitamins. Things that are lipophobic, so afraid, phobia of lipids, are going to have channels to go through, right? They can't directly diffuse across a membrane, but if you've got an open channel for that thing, it can still just passively move down a gradient. Otherwise, we're going to have to maybe do a little bit of stuff. So we'll see some movement of things through transcytosis. And particularly, this is how we'll see some proteins being exchanged. And in the brain, we'd have special types of mediated transport happening to move stuff across capillary walls. Okay. We are mainly going to be focusing on the exchange of water in this unit, though. Okay. So we're going to be thinking about the bulk flow of fluid across our capillary wall as we think about our capillaries. Okay. So we're thinking about the plasma and the stuff in it, but not proteins because they're too big and they're definitely not going to be able to cross a phospholipid bilayer. Okay. So we're going to see movement across our capillary walls, particularly of fluid, some stuff dissolved in it. Okay. The overall purpose is to distribute our extracellular fluid appropriately, distribute the contents of that extracellular fluid appropriately. And we're going to be thinking in detail about the pressures and the reasons we see changes here. So what I'm going to tell you right before we go, right, the first thing that you want to focus on getting down for sure, as we start our discussion of net filtration pressure, which is one of the real big things that we're thinking about when we think about capillaries, you want to think about the difference between filtration and absorption. So these are going to be direction words that you're focusing on. Okay, so I'm going to put some tissue up here. This is my little capillary. Okay, so we're thinking about filtration, so filtering stuff out of the blood, filtration. And we're going to be thinking about absorption, so moving into the capillary from the interstitial space. And we're going to see each of these things happen at different times due to different pressures. So that's what we'll be talking about next week. All right. So that's us for today. You got a quiz up on this stuff for Monday.